Welcome to the stem cell module, and in this talk we're going to look at induced pluripotent stem cells and their applications. So to begin, we're going to learn about how to study development and disease in humans. Now, to study any phenomenon, an appropriate experimental model is necessary. So if you want to study about human development, then we need an appropriate model that tells us information about human development. Now, for a long time, development in humans was studied in other model organisms, and then those mechanisms were assumed to occur in humans as well. However, recent advances in technology have enabled us to culture stem cells and study them to understand human development better. Now, in this case, the stem cells that we are culturing are of human origin, and that is why we're able to understand human development better. We're also able to use stem cells in order to treat diseases, and we're going to see how that is done in the next few slides. Now, this is where the induced pluripotent stem cells come into play. Now, nearly any cell in the adult body can be made into an induced pluripotent stem cell that has the pluripotency of an embryonic stem cell by adding 4 transcription factors to these adult cells. So as shown in the figure, if 4 transcription factors like OCT3 or 4, c-MYC, SOX2, and KLF4 are introduced into adult cells, those cells can be induced to become pluripotent stem cells. Now, the 4 transcription factors that were listed in the previous slide are master regulator transcription factors. SOX2 and OCT4 can activate another transcription factor called NANOG that establish pluripotency and block differentiation. Another transcription factor that is a master transcription factor is c-MYC. Now, c-MYC is known to open up the chromatin and make genes accessible to transcription factors like SOX2, OCT4, and NANOG. Finally, KLF4 is known to prevent cell death. Thus, these 4 master regulator transcription factors can induce pluripotency in differentiated adult cells. Now, many cell lineages and even entire embryos can be generated from single induced pluripotent stem cells. However, induced pluripotent stem cells are best at generating the cell types of the organ from which the parent somatic cell originated. Now, when we look at this figure below, basically shows this concept that we just talked about, where the induced pluripotent stem cells can form different types of cells, like they can form mesoderm lineate cells like red blood cells, or ectoderm lineate cells like neurons, or endoderm lineate cells like pancreatic cells, and thus they can give rise to a wide variety of cells. However, not all induced pluripotent stem cells are the same, and they may retain an epigenetic memory of their past, and this should be taken into account while studying the induced pluripotent stem cells. Induced pluripotent stem cells can be used for various medical applications. So, researchers have used patient-specific induced pluripotent stem cells for studying disease pathologies. Induced pluripotent stem cells can be combined with gene therapy to treat diseases as well. Induced pluripotent stem cell-derived progenitor cells that have been made from specific patients can be used in transplants in that same patient without the complication of immune rejection. Finally, differentiated cells derived from patient-derived induced pluripotent stem cells can be used for screening drugs. Let us look at one of the applications that we listed in the previous slide. Now, as sometimes it is difficult to study diseases if proper models are not available. One example is that of cystic fibrosis. Cystic fibrosis is a disease that we see in humans, And while there are mice models available for cystic fibrosis, the mouse models do not show the disease pathology the same way as we observe that in humans. So now what researchers are trying to do is they're trying to use induced pluripotent stem cells to produce models for studying cystic fibrosis in humans. For example, researchers made induced pluripotent stem cells from a person who has cystic fibrosis and then induced those stem cells to become lung epithelium, which is what is shown in the figure. These lung epithelium that were found showed the characteristic of human cystic fibrosis. And thus, by being able to create such a model in the lab, they can now study it and try to understand the disease better. A way in which we can combine gene therapy with patient-specific immunopluripotent stem cells to treat diseases is shown in this slide. Now, some diseases arise due to genetic defects. So the hope is that if the genetic defect is corrected by, let's say, gene therapy, then we can actually treat the disease. So researchers did such an experiment in a mouse model where they had a mouse model that was showing sickle cell anemia. And this was due to a mutation in its hemoglobin. So what they did was they obtained fibroblasts from the mouse, which are differentiated adult cells, so they're not stem cells. But then they were able to introduce the transcription factors OCT4, SOX2, KLF4, and c-MYC. This induced the fibroblasts to become induced pluripotent stem cells. Then what was done was, using these induced pluripotent stem cells, gene targeting was performed in order to correct the mutation that was present in the hemoglobin that caused sickle cell anemia in the first place. After those gene targeting experiments, the induced pluripotent stem cells whose hemoglobin gene was corrected were then isolated. And then they were allowed to differentiate into embryoid bodies so that they could eventually form hematopoietic progenitor cells. These hematopoietic progenitor cells were then transplanted back into the mouse, and it was observed that the sickle cell anemia was no longer present. And thus, in this way, we can use gene therapy along with induced pluripotent stem cells to treat diseases, at least in mice models, and now researchers are trying to apply this in human models. Now, many diseases can be caused by the actions of many different genes, and hence they are multigenic. Examples of such diseases are autism and muscular dystrophy. Induced pluripotent stem cells can aid in understanding multigenic diseases and provide therapies for treatment. An example of such a strategy is in the case where an induced pluripotent stem cells were formed from a patient that had autism. Now in this case, the induced pluripotent stem cells from the patient were then induced to form neurons. And when these neurons were studied, the scientists found that there was a mutation in a calcium channel protein called TRPC6. Now, scientists knew that there already was a drug available to correct these calcium channel defects. And so the cells could respond better when they were treated with this drug, which was called St. John's wort. And so finally, as a conclusion to this study, the patient's medication was altered to include St. John's wort as part of the medication regime. And thus this way, induced pluripotent stem cells can help us understand diseases and provide personalized medications. Pluripotent stem cells can also be used to rebuild tissues because they make these structures called organoids. Organoids are structures that possess many of the multicellular hallmarks of human organs. Now, organoids can be formed from both human embryonic stem cells as well as induced pluripotent stem cells. As shown in the figure, the pluripotent stem cells can undergo differentiation to form these different organoids as shown here, like the intestinal organoid, a kidney organoid, cerebral organoids, or optical cup organoids. Now, organoids mimic embryonic organogenesis, and hence they can also be used to understand organogenesis in humans. The pluripotent stem cells can self-organize into aggregates. So when we look at the figure, we have our pluripotent stem cells, and the pluripotent stem cells can organize themselves based on their differential adhesion, and the cells can start sorting out into the different proper positions. After that, lineage commitment of the cells can occur, and then the cells can differentiate to form a certain organoid. Let us look at how a cerebral organoid is produced. Now, in the case of producing a cerebral organoid, we normally will start out with the human pluripotent stem cells, as shown in the blue box in the figure. These pluripotent stem cells can then go ahead and form these embryoid bodies. Now, pluripotent stem cells will organize in the cell culture media, and they tend to form small spherical clusters, and these clusters are called as the embryoid bodies. Now, the embryoid bodies can then undergo changes to form as a neuroectoderm. When the neuroectoderm is then attached to a matrigel, which is a type of a solid support, then they can form these neural epithelial linings, which is what is shown in the large blue box. Now, this expanded neuroepithelium was added into a spinning bioreactor, and this enhanced the nutrient uptake of the cell mass, and ultimately they were able to form cerebral tissues. Thus, this is a way that we can actually start forming tissues from the pluripotent stem cells. Now, cerebral organoids have actually been used to understand diseases, especially neural diseases. Now, an example is where an induced pluripotent stem cell derived cerebral organoids from a patient with microcephaly were studied. And what was found is that there was a mutation in a gene that was important for symmetrical cell divisions in stem cells. So if we look at the figure, on the top part of the figure, we're looking at a control person who does not have microcephaly, or basically whose brains are the normal healthy size. And at the bottom panel, we're looking at a patient who has microcephaly, which basically means that the brain size is smaller than average. When we look at the cerebral organoid that was formed through induced pluripotent stem cells that were obtained from the different individuals, in the case of the control healthy individual, the cerebral organoids shows the presence of neurons that are shown in green and dividing cells that are shown in red. When we compare the architecture of the cerebral organoid of the control patient with the patient who is suffering from microcephaly, what we can see is there are many neurons present. However, we don't see as many dividing cells. What was found was that the patient suffering from microcephaly had a mutation in one of its genes. And this mutation causes premature neuronal differentiation, and thus we see many neurons present in the brain. However, the mutation also caused a decreased stem cell pool. And because of that, there weren't many stem cells that were present to proceed on to the formation of proper brain development. Thus, cerebral organoids help us to understand neural diseases. With this, we come to the end of our talk where we learned about induced pluripotent stem cells and the major medical uses with induced pluripotent stem cells.